So in a sealed cave in New Mexico, isolated from the surface for over 4 million years, researchers recently found something that honestly should have been impossible. They found ancient bacteria.
Right, but I mean, that wasn't even the impossible part, really.
No, exactly. The impossible part was that these bacteria, which, had never encountered a human being, let alone a modern hospital, they were already completely immune to our most advanced modern antibiotics.
It's just wild. It completely shatters the timeline we usually have in our heads about human medicine.
It really does. So welcome to today's deep dive. We are taking a massive stack of microbiology research, historical clinical data, and some really cool evolutionary biology case studies. And we're looking at this microscopic arms race happening right under our noses and, well, inside our bodies.
Yeah, the mission today is to figure out exactly how bacteria become resistant to antibiotics in the first place, and to really explore the rise of what people call superbugs.
Right. And if you've ever been prescribed, like a Z-Pack, or had routine surgery, this is going to fundamentally change how you view your own medicine cabinet.
Absolutely. But to understand that arms race, we really have to start with a very strict baseline.
Okay, lay it on us.
Because in clinical biology, resistance isn't just some vague philosophical concept. It is a precise operational laboratory measurement.
Right.
When a lab tests a bacterium, they're looking for something called the minimum inhibitory concentration, or the MIC.
Which is essentially like the absolute lowest concentration of a specific drug that visibly stops bacterial growth in a test tube, right? You just keep diluting the drug until the bacteria start multiplying again.
That's exactly it. And then comes the critical step. They compare that MIC against a breakpoint.
A breakpoint.
Yeah, a breakpoint is a literal threshold set by human standards bodies,
Yeah.
like the Clinical and Laboratory Standards Institute. If the MIC sits above that human-drawn breakpoint line, the organism is classified as resistant.
And if it's below, it's susceptible.
Exactly. But when you look at the clinical data, this is where that comforting illusion of medical precision kind of starts to fall apart.
Because those breakpoints move, right? Like the human drawn line changes as we gather more clinical data.
Yeah, meaning a strain of bacteria that was classified as totally susceptible to a drug in, say, 2005 might be reclassified as resistant in 2020.
Wow. And the fascinating part is that the bacterium itself hasn't changed at all. Just our human definition changed.
Right. It really highlights the friction between clinical resistance, where a drug just fails to cure a patient at a safe dose, and microbiological resistance, where the bacteria actually possesses a biological mechanism to fight the drug, even if we can technically still overwhelm it with a massive toxic dose.
Okay, let's unpack this, because before we even get to modern hospitals and these massive doses, we have to look way back.
Right, back to the cave.
Yeah, because my assumption going into this research was always that antibiotic resistance is a modern human-caused problem. I figure, well, we invented antibiotics in the 20th century, so obviously we created the resistance to them.
It makes intuitive sense, but yeah.
But that New Mexico cave story, plus I think they found genes in like 30,000-year-old permafrost too, it proves that is radically wrong.
We often forget what antibiotics actually are. I mean, they aren't synthetic inventions we just dreamed up in a vacuum.
Right.
They are mostly naturally produced by soil fungi and other environmental bacteria. It's ancient microscopic chemical warfare. These microbes have literally been fighting over limited resources in the dirt for billions of years.
So resistance isn't a human invention at all.
Not at all. It is simply the microbial world's ancient defense system against that natural warfare.
Basically, humans just stumbled into a microscopic war that was millions of years old, found the weapons, and started mass-producing them in factories.
That's a great way to put it. Which brings us to a pivotal moment in biology. Back in 1943, there was this massive debate about how bacteria actually survive these weapons.
Okay.
The question was, does exposing bacteria to an antibiotic actually cause them to mutate and become resistant?
Wait, like a Lamarckian adaptation, like the old idea that a giraffe gets a long neck just by trying really hard to stretch for high leaves and then passes that stretched neck to its baby.
Exactly like that. That was genuinely a prevailing theory at the time. The idea was that the drug induces this panic state and the bacteria somehow learn to fight back. Or there was the alternative theory. Do the genetic mutations happen completely randomly beforehand, and the drug just kills off everything except the ones that randomly mutated?
Enter the Luria-Delbruck fluctuation test.
Yes. Salvador Luria and Max Delbruck. They used E. coli to settle this, and it actually won them a Nobel Prize.
because they proved statistically that it's the second option, right? The drug doesn't cause the mutation. The mutations are already there.
Right, because bacterial populations are just incomprehensibly huge. A single infected wound can harbor 1 billion bacteria, and they have a random mutation rate of about 1 in every 100 million to 1 billion cell division.
Okay, so just by pure mathematical chance, in a population of 1 billion, you're always going to have a few mutants floating around.
It's exacto.
I was trying to picture this, and it's like, okay, the antibiotic is a brutal bouncer at a club.
Oh, I like that.
Yeah, so the bouncer doesn't walk down the line handing out VIP passes to let people stay. He just comes out and ruthlessly kicks everyone out of the club, except for the one random person who already had a VIP pass sitting in their pocket by complete accident.
That is exactly what happens. And what's wild about that bouncer effect is what happens next. The antibiotic acts as a sieve, a filter.
Right, it applies massive selective pressure.
Yes, all the susceptible cells die, leaving that rare, pre-existing resistant variant alive. And because all of its competition for food and space is suddenly gone...
It just rapidly repopulates the entire area with its resistant clones.
Exactly. And what's fascinating here is that Alexander Fleming, the guy who discovered penicillin, literally warned us about this sieve effect.
Wait, really? The guy who discovered it?
Yeah. In his 1945 Nobel Prize speech, he explicitly said that the man who is careless with penicillin would actively select for these resistant strains. He saw it happening in his own lab when he used sublethal doses.
So he essentially mapped out Darwinian selection playing out over hours instead of millennia.
He did, and he was completely vindicated. Within a decade of mass-producing penicillin during World War II, penicillin-resistant staph infections were already sweeping through hospitals.
Man, okay, here's where it gets really interesting for me. If the resistance is this pre-existing VIP pass, what exactly does that pass allow the bacteria to physically do?
Right, the actual mechanism.
Yeah, like when you swallow a pill and the antibiotic enters your bloodstream, how do the resistant bacteria actually fight it off? The research outlines 5 core biochemical strategies, and it honestly reads like an escalating warfare manual.
It really is an arms race, and nearly every resistant infection a doctor will ever encounter uses one or more of these 5 mechanisms.
Okay, let's go through them. Mechanism number 1, enzymatic destruction. I think of this as the bomb squad.
The bomb squad, yeah. The bacteria literally produces a specialized enzyme that chemically destroys the antibiotic before the drug can even do its job.
And the classic textbook example here is beta-lactamase, right?
Exactly. Penicillin and drugs like it rely on a specific molecular structure called a beta-lactam ring. It's the core key that makes the drug work.
Okay.
The bacterial enzyme beta-lactamase basically acts like molecular scissors. It just goes in, snips that ring open, and renders the drug totally inert.
And this is exactly why my doctor gives me Augmentin for a sinus infection, because Augmentin is a combination drug. It's a moxicillin, but paired with clavulanic acid.
Right, and what does the clavulanic acid do?
Well, from what I read, it doesn't even kill bacteria. It's just a biochemical bodyguard. It jams up the beta-lactamase bomb squad so the amoxicillin can actually slip past and work.
You've got it. But then the bacteria escalate. What if the bomb squad gets bypassed? They move to mechanism 2, target modification.
Right. Because antibiotics are highly specific. They're designed to bind to very specific proteins on the bacteria, like a key fitting into a lock to stop them from, say, building their cell wall.
So the bacterium just mutates the lock. It alters the 3D shape of that target protein just slightly. The protein still does its normal job of building the cell wall, but the antibiotic key no longer fits into the docking port. The drug just bounces off.
This is what MRSA does, right? Methicillin-resistant Staphylococcus aureus. I was reading. It carries a specific genetic instruction called the MECA-DO gene.
Yes, the MECA gene.
And that changes its docking port so fundamentally that entire classes of standard drugs just become useless.
Which forces doctors to use different, often much harsher drugs. But what happens if the drug does manage to get inside the cell? That brings us to mechanism 3, efflux pumps.
This one is wild to me. It's like a submarine taking on water, and they just turn on the bilge pumps.
That's exactly it. The bacteria evolve physical, membrane-embedded pumps that actively capture the antibiotic molecules and spit them right back out of the cell as fast as they enter.
They burn their own cellular energy to do it, just ensuring the drug never reaches a lethal concentration inside.
Right. Pseudomonas aeruginosa, which causes severe hospital-acquired pneumonia, uses this heavily. It's an absolute nightmare in intensive care units because you are pumping the patient full of drugs, and the bacteria are just pumping them right back out.
So frustrating. So mechanism 4 is reduced permeability. Because why bother burning energy to pump it out when you can just lock the doors?
Exactly. Some bacteria have an extra outer layer of armor.
These are gram-negative bacteria, like Klebsiella.
Yes. Gram-negative just means they have a secondary outer cell membrane, a double-walled defense. To get through that outer wall, antibiotics have to travel through microscopic pores called porin channels.
And the bacteria can simply mutate to shrink those channels
Okay.
or close them entirely.
Yeah. Which leaves us with the fifth mechanism, metabolic bypass, the detour. If an antibiotic successfully gets in, finds its target, and successfully blocks a specific enzyme that the bacteria needs to survive, well, the bacteria just evolves an entirely alternate biological pathway.
It builds a detour that does the exact same job, but is totally invisible to the drug. It completely routes around the blockade.
We see this constantly with resistance to sulfonamide antibiotics. The bacteria literally just invent a new way to synthesize the folic acid they need to survive.
Okay, but I have a major biological question here. We established earlier with Luria delbringt that these mutations happen randomly, maybe 1 in 100 million cell divisions.
Yes.
If a random mutation is that incredibly rare, how on earth are we seeing entirely different species of bacteria becoming resistant to 5 different drugs so incredibly fast? I mean, evolving a brand new pump or a new enzyme should take thousands of years.
That is the crucial missing piece of the puzzle. Bacteria do not just pass these survival traits down vertically to their offspring, they trade them horizontally with their neighbors.
Oh, wow.
It's called horizontal gene transfer.
Right, so they literally trade DNA. They don't have to wait for evolution. They just hand the physical blueprints to the cell next to them.
It is remarkably efficient. There are 3 main ways they do this. The most prominent is conjugation.
Which is what, exactly?
2 living bacteria physically connect. They build a microscopic bridge between themselves called a pilus. Through that tube, they transfer plasmids.
Plasmids.
Yeah, plasmids are little mobile circles of DNA that exist separately from the main chromosome. And the scary part is they can carry multiple resistance genes at once.
So it's not even just one gene at a time. It's a whole care package of resistance.
Exactly. Then there's transduction, where a bacteriophage accidentally packages up some bacterial DNA and injects it into the next cell it attacks.
Right.
And finally, transformation, where a living bacterium just scavenges naked DNA fragments floating around from dead bacteria and incorporates it into its own genome.
So wait, they aren't just evolving slowly over millions of years like Darwin's finches on an isolated island. They are tossing genetic cheat codes across the hallway to completely different species of bacteria.
They absolutely are. And understanding this changes everything about how we view infectious disease. To really grasp how fast horizontal gene transfer is, you have to contrast it with Richard Linsky's famous long-term evolution experiment.
Oh, I know this one. His lab has been breeding E. coli every single day since 1988, right?
Right. They've tracked over 75,000 bacterial generations over decades. It's a beautiful demonstration of slow, spontaneous mutation.
Yeah.
After tens of thousands of generations, one single population finally evolved a rare, complex ability to metabolize citrate. It took decades of purely vertical parent-to-offspring evolution to get one novel trait.
But with horizontal gene transfer, they skip the decades of waiting. They just connect a tube and download the citrate-eating blueprint or the antibiotic resistance blueprint from the bacteria next door in 5 minutes.
And that speed is why the global spread of superbugs is so terrifying. Look at the emergence of CRE.
Carbapenem-resistant enterobacteriation.
Yes. Enterobacteriaceae is a huge family of common gut bacteria, normally totally harmless. But carbapenems were our heavy hitters, our last resort antibiotics.
Right.
In 2008, an isolate was found in a patient hospitalized in New Delhi carrying the NDM1 gene, which produces an enzyme that totally crushes carbapenems.
And because that NDM1 gene was riding on one of those mobile plasmids, it didn't just stay in one species.
No.
It crossed into different species of gut bacteria and circled the globe via international travel and hospital transfers in just a few years. It turned normal gut bacteria into unstoppable pathogens.
So knowing all this, knowing they have this biochemical arsenal and they can share blueprints instantly, we really have to look at how human behavior is accidentally helping them win.
Well, I was reading about this idea of a fitness cost. If running all these extra bilge pumps and building all these complex enzymes takes energy, won't the resistant bugs be slower and weaker than normal bacteria?
That's the logical thought, yeah.
Right. The assumption was always, if we just stop using a specific antibiotic for a few years, the resistant bugs will be outcompeted by normal bugs and die out.
And that was a huge hope in evolutionary biology for a long time. But nature is endlessly stubborn. What researchers found is that while there is an initial fitness cost, bacteria quickly acquire compensatory mutations.
Meaning what? They tweak their machinery to make it cheaper to run?
Precisely. They develop secondary genetic mutations that optimize the resistance mechanism. They restore their full health and growth speed while keeping the resistance gene.
Oh, wow.
So the resistance becomes essentially zero cost to the bacteria, which is terrifying because it means the resistance persists indefinitely, even if we stop using the drug entirely.
Which brings us to the massive debate around agriculture. For decades, a huge percentage of all antibiotics manufactured globally weren't even going to sick humans.
No, they weren't.
They were being fed to livestock, pigs, cows, chickens at low subtherapeutic doses every single day just to promote faster animal growth.
Which is the exact sublethal selective pressure Alexander Fleming warned us about.
Right.
You are constantly running that sieve, filtering for resistant bugs in the guts of millions of animals. The EU banned this practice outright in 2006, and the FDA has progressively restricted it in the US, but it remains a heavily debated global issue.
Because those zero-cost resistant genes don't stay on the farm. Through water runoff, meat processing, and farm workers, they jump into the human microbiome.
Exactly.
Okay, speaking of the human microbiome, I want to ask about something that completely broke my brain in this research.
Oh, let's hear it.
I have been told my entire life, by pediatricians, by public health campaigns, literally by my own mother, that I must always finish every single pill in my bottle of antibiotics.
Right, the golden rule.
Yeah, the logic was always, if you stop early just because you feel better, you'll leave the strong bacteria alive and you'll create a superbug.
Are the data really saying that's a myth? This is honestly one of the most significant and controversial paradigm shifts in modern medicine. A highly influential 2017 commentary in the British Medical Journal by Martin Llewellyn and his colleagues synthesized a growing body of evidence arguing that the old rule is often fundamentally wrong.
Explain the mechanics of this because it feels like medical heresy. Why would stopping early be better?
Think back to the sieve effect we talked about. The antibiotic doesn't create the mutation, it selects for it.
Okay.
Imagine you have a bacterial infection. You take antibiotics, and after 5 days, your clinical symptoms are totally gone. The bacterial load has dropped so low that your own immune system now had the upper hand.
Right, the infection is basically neutralized.
But continuing to pump antibiotics into your body for another 9 days doesn't help cure you any faster. What it does do is provide unnecessarily prolonged exposure to your entire body. You are keeping the sieve running longer in your gut. You are applying more selective pressure to the surviving innocent bystander bacteria in your microbiome, giving them more time to enrich those resistant populations.
So, stopping the drug when the infection is clinically resolved might actually be safer in many cases because you are turning off the evolutionary sieves sooner.
For many common infections, yes. Now, I want to be very clear here. This heavily depends on the specific infection.
Right, of course.
Things like tuberculosis require incredibly long, strict courses. You should always follow your personal doctor's advice, but broadly speaking, at the public health level, the settled message of always finish the course no matter what is being aggressively revised.
Wow. So what does this all mean for you, the listener, in your everyday life? We've zoomed in on plasmids and enzymes, and we've talked about farm policies, but let's zoom out to the real-world stakes.
Yeah, let's bring it back.
First, let's address the word superbug. When I hear that, I picture, like, a glowing, radioactive comic book monster.
And that's a really dangerous misconception. A superbug is not a newly discovered organism with a terrifying new superpower.
It's not.
No, it is usually just a completely ordinary bacterium like the E. coli that naturally lives in your gut right now that has accumulated multiple different resistance mechanisms.
Okay.
It's called MDR, or multi-drug resistant. It's just a matter of degree. It has a pump and a modified target and a destructive enzyme.
But the stakes of those ordinary-looking bugs accumulating those powers are massive.
Yeah.
I saw the World Health Organization views antimicrobial resistance as one of the top global public health threats.
Oh, absolutely.
Because it's not just about getting a bad cough that's hard to shake. Modern medicine is literally built on a foundation of antibiotics.
Without reliable antibiotics, routine surgeries become life-threatening gambles. Chemotherapy, which suppresses the immune system, becomes incredibly dangerous because patients can't fight off background infections. Organ transplants, nearly impossible.
And when the standard drugs fail, doctors are forced to dig into the historical archives. I was reading that they actually have to dust off older last resort drugs that were abandoned in the 1960s because they were too toxic.
Yes, drugs like colistin.
Right. You can cause severe kidney damage. But if you have a multi-drug resistant infection, doctors have no choice. They have to use it.
Which is exactly why the concept of antimicrobial stewardship is so critical today. It's a clinical movement focused on prescribing antibiotics only when absolutely necessary at the precise dose and for the shortest effective duration to really slow down this evolutionary selection process.
And that ties into the One Health framework, right?
Yes, the recognition that human health, animal health, and the environment are all profoundly interconnected.
Okay, there is one last major misconception I want to forcefully clear up before we wrap. And it's something I hear people say all the time when they have a cold.
I think I know what you're going to say.
They say, oh, I try not to take antibiotics because I don't want my body to become resistant to them.
That is biologically impossible.
Right. Your body does not become resistant. The drugs do not interact with your immune system. They don't train your human cells. They act entirely on the microbial population inside of you.
Exactly. It is the bacteria that evolve, not you.
So when you take an antibiotic, when you don't need one, like for a viral cold, where the drug literally does nothing to the virus, you're just carpet bombing your own microbiome and selecting for resistant bacteria that might infect you or someone else later.
It's the ultimate communal tragedy. An individual's choice to misuse an antibiotic seems inconsequential to them, but it really contributes to a population-level disaster.
Man, we have covered a massive amount of ground today. We started millions of years ago in a dark, sealed cave in New Mexico looking at ancient chemical warfare.
We did.
We explored Luria and Delbruck's 1943 experiment that proved mutations are random and pre-existing. We broke down the incredible bacterial arsenal, the bomb squads, the pumps, the locked doors.
Skull playbook.
We saw how bacteria share blueprints across species, and we unraveled the shifting medical guidelines around prescriptions. Thank you for navigating this incredibly dense landscape of bacterial warfare with me.
Ugh, it's a complex topic, but it's so essential to understand if we are going to keep our medical system functioning. However, before we go, this raises an important question. And there's one more concept in the research that we didn't fully explore today, and it kind of challenges everything we just talked about.
Wait, really? Lay it on us.
Well, we've spent this entire deep dive talking about genetic resistance, bacteria mutating to actively fight off antibiotics, producing enzymes or building pumps, right?
Yeah.
But researchers are increasingly studying something called tolerance and persister cells. They are finding populations of bacteria that don't have any genetic mutations for resistance. They don't fight the drug at all.
Then what do?
Instead, they just shut down.
Shut down, like they turn off.
Exactly. They form structured communities called biofilms, and the persister cells inside simply slow their metabolism to a halt. They go completely dormant.
Wow.
And because almost all antibiotics rely on the bacteria actively growing and dividing in order to kill them, the drug just washes over them harmlessly. They just wait us out.
Wow.
When the antibiotic is gone, they wake back up and resume the infection. So it raises a profound question for you to explore next. If the future of treatment failure isn't just about bacteria mutating to fight back, but bacteria simply going to sleep, how on earth do we treat a disease that plays dead?
That is haunting. It really brings us back to that broken medical expectation of precision we talked about at the start. Just when you think you have the picture clear, the biology finds a way to blur the lines again. Thanks for joining us on this deep dive. Keep questioning the microscopic world around you, and we'll catch you next time.